t-RNA. Transfer RNA played dual role. It read the codons present on the mRNA. It transfer amino acid from cytosol to protein synthetic area. Two functions it carries. Other name to t-RNA. Other names to t-RNA. Adapter RNA. Smallest RNA. Soluble RNA. Adapter RNA. Adapted to mRNA. Attached to the mRNA. Translate the codons present on the mRNA. So it is also called translator RNA. Smallest RNA. The molecular weight is 25,000 Daltons only. It consists the 80 to 90 nucleotides. And the molecular weight is 25,000 Daltons only. So it is the smallest RNA and it is soluble in 1 molar sodium chloride solution. So it is soluble RNA. Function: translate or the decode the codons present on mRNA. Transfer amino acids from cytosol to protein synthetic area, that is on the ribosomes. Clover leaf model. Model to t-RNA proposed by Hawley. Clover leaf model to the t-RNA proposed by Hawley. According to clover leaf model, a t-RNA consists 5 arms, 3 loops. But actually, the shape of t-RNA is inverted L shape. Inverted L shape. Actual shape of t-RNA is inverted L shape. Clover leaf model to the t-RNA that is proposed by Hawley. According to this clover leaf model, 5 arms, 1, 2, 3, 4, 5 arms, 3 loops, 1, 2, 3 loops present. At one end, 3 nitrogen bases present freely, and the 3 nitrogen bases are CCAN. Where it is present? At 3' end. CCA, cytosine, adenine. These 3 nitrogen bases present freely at the 3' end, so it is also called CCAN. Here, amino acid attaches. So it is also called amino acid binding site. CCAN or 3' end is amino acid binding site. One loop recognize ribosome recognizing loop. One loop recognize enzyme. Activates amino acid. Amino acid activating enzyme recognition loop. And another loop that present quite opposite to CCN. Here also 3 nitrogen bases present freely, called anticodon. That is called anticodon loop. Anticodon loop. Now, ribosome recognizing loop, also called DHU loop. Because it consists an unusual nitrogen base, dihydrouridine. So it is also called DHU loop, that is full form dihydrouridine loop. So according to cloverleaf model, it was proposed by Hawley, 5 arm, 3 loops present. One arm, that is the accessory arm, function not known. At one end of the one arm, CCA, 3 nitrogen bases present freely. That is present at the 3' end. Here amino acid is attached, so it is also called amino acid binding site. Amino acid binding site is CCAN or 3' end. So in between the amino acid and adenine present in this CCAN, a bond is formed, that is covalent bond. Covalent bond in between amino acid and CCAN formed in between carboxyl group of amino acid and OH group of adenine. A covalent bond is formed in between carboxyl group of amino acid, OH group of adenine. That means amino acid attached to adenine present at the 3' end with the help of a covalent bond. One t-RNA carries one type of amino acid only. 20 types of amino acid recognized by 61 types of sense codons. So total types of t-RNAs involved in protein synthesis, 61 types. Type of t-RNA based on type of anticodon. Type of t-RNA based on type of anticodon. Total types of t-RNA 61 and the type of codon depend on type of anticodon. 61 sense codons, 61 anticodons. So types of t-RNAs not present in cytoplasm or cytosol. For example, nonsense codons UAA, UAG, UGA. UAA, UAG, UGA. These are the nonsense codons. A, U, U, A, U, C, A, C, U. So t-RNAs with anticodons AUU, AUC, ACU are absent. t-RNAs with anticodons AUU, AUC, ACU absent. t-RNA that carry amino acid is the first codon, initiator t-RNA. Initiating codon AUG that recognize methionine. So t-RNA which carry first amino acid or initiator t-RNA that carries the anticodon. AUG is the codon, so anticodon is the UAC. So anticodon of initiator t-RNA is UAC. t-RNAs with anticodons AUU, AUC, ACU are absent. Next one, translation. Transcription, types of RNA is completed. And the next one, translation. Based on the genetic information present on the mRNA, formation of the polypeptide chain or the synthesis of the polypeptide chain in accordance to genetic information present on the mRNA. that is called translation. Decoding of the genetic information present on the mRNA by tRNA, that is called translation. In eukaryotes, translation occurs in cytoplasm. In translation, proteins are, the polypeptide chain is formed or proteins are formed.